When was the last time you hit the gym or went for a run? Exercise has been synonymous with looking good. I mean, that's a huge reason why people work out, right? But consider the benefits exercise have on our brains. Research has shown that aerobic exercise in rats led to increased hippocampal volume, neurogenesis, and long-term potentiation. All these processes form the foundation of memory and learning. So how exactly does physical activity translate to better memory? Demonstrated in rats that were put on a 30-day running regime, exercise led to increased transcription of a number of transcription factors that ultimately upregulate the expression of a very key protein in the hippocampus, BDNF. BDNF, or brain-derived neurotrophic factor, is a neurotrophic growth factor, meaning that it is an important regulator of growth, survival, and differentiation of new neurons and synapses. It plays a critical role in long-term potentiation, or LTP. LTP is the molecular model of long-term memory formation. It is a series of changes to the synapses of two neurons following repeated stimulation. The result is a long-lasting enhancement in signal transmission between the two neurons, conferring a stronger synapse and ultimately a stored memory within the brain. To understand LTP, let's take a closer look at a synapse. Many receptors are present on the post- and presynaptic membrane. When high-frequency action potentials reach the presynaptic dendrite, it triggers the release of a lot of the neurotransmitter glutamate. When a lot of glutamate binds to the mPAR receptors on the postsynaptic neuron, a huge influx of sodium ions results, which in turn triggers the influx of calcium ions through neighboring NMDA receptors. Calcium ions act as important secondary messengers, indirectly leading to the increase in mPAR receptors in the postsynaptic membrane. The result is a greater depolarization in response to lower frequency action potentials. The synapse is said to be stronger. Back to BDNF. Other than mPAR and NMDA receptors, there are also TRKB receptors on the synaptic membranes. BDNF binds to these receptors, activating them. As TRKB is a receptor tyrosine kinase, this initiates major phosphorylation cascades of the signaling pathway similar to that of insulin signaling. Activation of one of the pathways leads to a rise of intracellular calcium ions, which upregulates translation of mPAR and NMDA receptor mRNAs. BDNF also induces the rapid surface translocation of mPAR to increase excitatory transmission. With more BDNF, LTP is improved, and so are our memory functions. Although it has been widely demonstrated in rats that aerobic exercise increases BDNF levels and improves spatial memory, we have yet to determine the effects of exercise on other more human types of memory, such as verbal memory. Regardless, exercise delivers many other advantages to our cognitive functions. So the next time you plan a study schedule, consider incorporating a short cardio exercise.